Greetings, I'm Lisette Morris, the Ingenuity Project Executive Director. I'm so thrilled to share with you this showcase of research presentations, a culmination of work by our juniors and seniors during the 2019 and 2020 school year. In a typical year, students showcase their work at our annual Math and Science Research Symposium in May. However, as a result of the COVID-19 pandemic, our in-person gathering needed to be converted to something virtual. In March, when schools closed in Baltimore City and around the country, our students conducting research had to put their work on hold. In many cases, students were unable to complete their final tests and develop conclusions of their projects. Having 1 to 2 years of hard work come to a halt was a big disappointment to their research projects. And yet, students rallied to record these incredible presentations. The Research and Innovation Practicum programs are a flagship component of the Ingenuity Project that supports students through a multi-year curriculum that culminates in independent research. This process begins with coursework in 10th grade to learn about the scientific and engineering research and explore personal areas of interest. Then, students seek out a mentor to design and execute their own research. Students have a unique opportunity to work alongside professional scientists, mathematicians, technologists, and engineers in leading institutions such as Johns Hopkins University, Morgan State University, and Towson University. We also have a growing list of STEM companies as well. This video showcase is now a keepsake for our students and our community, and something I'm personally excited to share with stakeholders who have not had a chance to see the rigorous and deep work Ingenuity students take part in during the practicum. In this video, you will see the work of our seniors who have had a chance to work in research labs for 2 summers and wrap up their project in the fall through their submission to the Regeneron Science Talent Search, the nation's oldest and most prestigious STEM competition for high school seniors. Our juniors began their research completing more than 120 hours in their labs during the summer of 2019, and they were working approximately 8 hours a week in their lab during the school year until businesses and schools closed in March. Thank you so much for your support, and may your mind be open to the impressive findings our students have made. Thank you very much.
Hi. My name is Alumbro, and I've been working with Dr. Karen Hyman at the Landscape Hydrology Lab in the Johns Hopkins Whiting School of Engineering to look into the water quality of Baltimore City streams. It's important to understand the content of our streams because this in return affects our environment and our water usage. This year, I've been fortunate enough to learn how to code and analyze large datasets as well as map on GIS. In my research experience, I've understood the complexity of using mathematical models to define a natural occurrence. Doing work in this field has taught me that there can be a lot of disturbances to a pool of water. However, it is our job to understand the relationship between our lifestyle and our environment.
Hello, my name is Ihemriorochi Amanze. I work at Johns Hopkins University in their Haptics Medical Robotics Lab, also known as HAMR Labs. I work most closely with their prosthesis department, which deals with implementing haptic feedback into prostheses. Haptic feedback is a sense of touch we experience every day, like when we decide something's hot or cold, or sharp or soft. So far, I have looked at how currently haptic feedback is implemented in our everyday life and how it is currently implemented in prostheses. My most recent project involves helping develop 2 different haptic feedback methods for a prosthesis that we also developed. We had users use these prostheses in the box and blocks test, which involves them moving blocks from one side box to another using the haptic feedback to decide which one is better for further use. Using this test, we decided on a prosthesis method to use in our further testing. Going forward, I would like to develop my own research project in which I will develop a prosthesis hand that will help the user analyze and identify an object in their hand using only haptic feedback. Thank you.
Hi. My name is Tate Bothner, and I'm a junior in the Ingenuity Research Practicum. Before COVID-19 shut schools and labs down, I was studying CRISPR systems in the Department of Biochemistry and Molecular Biology at the Johns Hopkins Bloomberg School of Public Health. In short, CRISPR systems are immune systems for bacteria, protecting bacterial cells from invasion by viruses. But why does CRISPR matter? Genetic disease affects millions of people worldwide. Most genetic diseases cannot be cured because they are the result of a mutation in a person's DNA, the long molecule that carries genetic information. With the discovery of CRISPR, however, it is likely that in the near future, scientists may be able to genetically edit humans in order to cure genetic diseases. Before this can happen, though, we need to understand all of the complex structures and processes that occur within CRISPR systems. In my lab, we study how CRISPR systems work on a fundamental level in bacteria. Understanding how the CRISPR systems work within a cell can allow us to better use the inner workings of a cell to our advantage. This has applications to future gene editing and gene targeting technologies. Thank you for listening.
Hi, my name is Eli Brody, and I conducted my research in the GUREC lab on the Johns Hopkins Homewood campus. The GUREC lab is a part of the Institute of Nanobiotechnology and focuses on learning more about cell functions through in vitro modeling. My research is focused on the biological basis and effects of Marfan syndrome, which is a genetic disorder occurring in about 1 in 10,000 individuals. The manifestations of Marfan syndrome include tall stature, hyperextensible joints, elongated digits, and the development of aortic aneurysms. An aneurysm is the weakening of the wall of blood vessel, and the rupturing of a wall of the aorta is the main cause of death in Marfan patients. The mutation which causes Marfan syndrome leads to a lack of production of fibrillin 1, a crucial structural protein of the extracellular matrix, also known as the ECM. The ECM is responsible for giving structure and elasticity to blood vessels. In the absence of fibrillin, the ECM loses elasticity, which is necessary for resisting the powerful pumps of the heart. This is especially necessary in the aorta, where the heart pump creates the most force. In order to model the effect of the heart's pressure on the blood vessels, I learned to fabricate microfluidic devices, which I will add Marfan endothelial cells to. These are used to model the small blood vessels within the body in order to understand the impact of stress on the vessel lining cells derived from Marfan patients. The goal of this research is to build a working model of the effects of stress imposed by blood flow on vessel lining cells to better understand the causes of aortic aneurysm.
Hello, my name is Riyan Campbell, and during my sophomore and junior year, I worked alongside my mentor, Dr. Vitolo, at the University of Maryland Greenbaum Center, and my research topic was exploring the structural phenotypes of microtentacles. Today, many do not know that, on average, 42,000 people die each year due to metastatic breast cancer. Sometimes, these cancer cells that come from our regular cells form finger-like protrusions, which help these cells spread to distant parts of the body, bringing the cancer with it. The goal of my project is to test the lasting effects of chemotherapeutic drugs on these finger-like structures called microtentacles. By doing this research, it could bring forth something truly amazing in terms of finally finding a cure to this global disease known as cancer that affects so many. Thank you.
Hi, I'm Sabina Celnik, and I've been conducting my research at the Dolan Lab, a part of the Brain Science Institute at Johns Hopkins. So an overarching goal of the Dolan Lab has been to find potential treatments for autism occurring in adults. Autism is a developmental disorder in humans, symptoms of which include difficulty in social interactions and difficulty with communication. At the Dolan Lab, we use mice as an animal model for studying autism. Mice are a good model for studying autism as, like humans with autism, they do not find social interactions rewarding, specifically in their adulthood. So a smaller goal in completing this longer goal has been to find a social behavior to determine whether the potential treatments for autism are valid or not. And we believe that social dominance could be this behavior. A commonly used assay in determining the dominant status of mice has been the tube test assay, in which 2 mice enter a tube and go head to head, and the mouse that pushes the other to the back of the tube is the dominant mouse. However, despite it being a commonly used assay, there have been conflicting results from different studies over the years, throwing into question the validity of the tube test assay. So my research has been entirely about fully determining whether or not the tube test is a valid assay for studying social dominance, so that we can use social dominance as the behavior to determine if the autism treatments are valid or not. In order to determine the validity of the tube test, I have been running the tube test assay and comparing it with another dominance assay that determines the dominance of mice, called the urine marking assay, in which two mice are placed into a cage separated by a perimeter, and whichever mice urinates closer to the barrier between the mice and more is the dominant mouse, as it's like asserting its territory over the other mouse. I've already run this experiment once, comparing the tube test assay to the urine marking assay, and I will be continuing my research by continuing to run the experiment and further determine whether or not the tube test is indeed a valid experiment for discovering social dominance.
Hi, my name is Seth Chng-lim, and I did my research at the Roberts Lab on Johns Hopkins Homewood campus. I studied AI, or artificial intelligence, a branch of science poised to make massive contributions to this world. One of the most important types of AI are neural networks, or computational algorithms created by scientists modeled loosely after the human brain. These networks can do many things, such as weather forecasting, medical research, experimental data validation, and risk management. Convolutional networks are a subclass of neural networks that are used for image recognition. A major issue with these convolutional networks are that they have trouble with image segmentation, a process of detecting objects within an image. Due to this, I worked on and created a neural network that can segment basic shapes from images. I developed a modified version of a neural network using a structure called a U-net to do the segmentation, and I trained the network using simulated data I generated computationally. Unfortunately, I haven't gotten around to testing my dataset or my network, so I hope to do that in the future.
Hi, my name is Tendai Coady, and I've been conducting research with the Marchioni lab. So when trying to determine how to treat cancer in a patient, it's helpful to know the cancer prognosis, which is the expected outcome. And this is important because it lets us know whether this patient requires more aggressive treatment strategies, such as chemotherapy, or not. And the current assays used for prognostication of breast cancer are far from perfect in terms of accuracy. So we sought to use machine learning with gene expression data to develop a better prognostic classifier. The classifier compares levels of gene expression between genes to determine the prognosis. And a huge potential problem with this is overfitting, basically the classifier using unnecessary details when trying to prognosticate, which can be a huge hindrance to the performance of the algorithm in determining prognosis. So to counter this problem, we made all of the decision rules in the algorithm follow under a specific biological logic. In other words, they relate logically to real cancer biology. And so the algorithms we've generated so far have been less accurate than currently clinically approved assays in the testing phase. So that means more research is required.
Hello, my name is Filipovitz, and I was fortunate enough to be able to work in the Cordis lab at Johns Hopkins University last summer in the school year. In lab, I researched the neurobiology of animal behavior. Behaviors are very difficult to track because of the complexity of the neuron pathways a behavior follows before being executed. In order to understand human behavior, it is necessary to first learn more about behavior as a whole and in general. This is made possible by studying organisms with less complex nervous systems. The Cordis lab uses both worms and spiders as models, both of which have significantly less neurons compared to a human. I specifically focused on working with the spiders. By tracking the limb movements of the web building process, I am able to see how behavior is developed over time and executed. I have been focusing on training artificial intelligence to track limb movements for us, allowing accurate results in a very efficient time manner. Thank you.
Hi, my name is Maddie Jaffe. I'm in 11th grade and I'm doing research at the Institute of Marine and Environmental Technology on the biodiversity of the Inner Harbor. So the big concept of my research is that you can't conserve what you don't understand. And before this project started, there was a huge knowledge gap in terms of what biodiversity and what species existed in Baltimore's Inner Harbor. And if you don't understand what species exist in the harbor or how humans influence them, you can't take any actions towards conservation of the community. So my research focuses on understanding how that community of biodiversity in the Inner Harbor changes over time and how environmental factors influence that community. Because a lot of times people assume that nothing can live in the Inner Harbor, that it's too polluted, but that's not true. There's actually a thriving ecosystem, and by understanding that ecosystem and how environmental factors influence it, we can understand how we impact the harbor and take action to conserve the species that live within it. So to do this, IMET had a partnership with the National Aquarium. We collected biodiversity samples from 2 sites in the Inner Harbor. One of them was near a floating island wetland, which simulated healthy wetland ecosystem conditions, and that is something that the National Aquarium created in hopes of tracking the biodiversity in the harbor and simulating just a healthier ecosystem. So that was one site where we tracked the biodiversity. And the second one, also in the Inner Harbor, was near a dock. So we had 2 different sites where we were collecting biodiversity samples of what lived in the harbor, one near this ideal ecosystem and one just in the regular harbor conditions. So we collected biodiversity using biodiscs. Biodiscs look kind of like CDs, and they are submerged in the water, and they build up communities of microscopic creatures over time. And then we can film these biodiscs and count the number of individual species on the discs in order to understand what is living in the harbor at a microscopic level. And this can include mudworms, barnacles, mussels, all that sort of thing. So we counted the number of species, and that allowed us to track from 2016 to 2019 what was existing at the harbor in these different sites and how those numbers changed over time. So specifically, I compared this biodiversity data to dissolved oxygen levels, because dissolved oxygen is something that marine species in particular rely a lot on, and if the dissolved oxygen level is too low, then you can have a low oxygen event that will trigger a die-off and kill a lot of species in the harbor. So I compared the biodiversity levels of the 2 sites to dissolved oxygen levels over time in order to see if the site near the wetland ecosystem that simulated healthier conditions was more resilient in the face of these low oxygen events. And what I found was, yes, they had a faster rate of return in biodiversity levels from these low oxygen events, which means that if you implemented more of these floating island healthy condition simulated ecosystems into the harbor, then you can make the whole ecosystem as a whole more resilient towards pollution in the harbor. So currently I'm looking at individual species in the harbor and seeing how they change over time. So before I was looking at the community as a whole and how it responded to dissolved oxygen, and now I'm looking at individual species and their role in the community over time, and also comparing those individual species to environmental factors. So this just contributes to what I was working on before. And hopefully this can all contribute towards making the harbor ecosystem a healthier place for the species that live within it.
Hi, my name is Young Lee. I work in Dr. Steve Farber's lab at the Carnegie Institution for Science, where I study lipid metabolism in zebrafish. The zebrafish is emerging as a key model for studying lipid metabolism due to its embryonic transparency, high reproduction, genetic similarity to humans, and genetic tractability. Cardiovascular disease is a leading cause of death worldwide. In particular, the prevalence of a high-fat diet in Western culture is an increasingly concerning health issue. Lipoproteins are the particles that transport the fats we consume throughout our body for use and storage. Studying lipoproteins and their underlying mechanisms will contribute to the development of therapeutics and treatments for cardiovascular disease. Previous studies have shown the decrease in lipoprotein size and increased number of them is linked to an increased chance of getting cardiovascular disease. ApoA4 is a gene that is known to play a role in regulating the size of lipoproteins. For my project, I looked into the effects of knocking out and upregulating ApoA4. My results have shown that the level of gene expression did not have a significant effect on lipoprotein size and quantity. This suggests that there may be other factors that are required for regulating lipoprotein particle size. ApoA4 has multiple paralogs, and in the future, I would like to analyze how those other ApoA4 genes affect the lipoprotein profile in zebrafish. Thank you.
Hi, I'm Sarah, and I'm conducting my research at the Cullen Lab at Johns Hopkins University. The Cullen Lab is a biomedical engineering lab that focuses their research on the vestibular system. The vestibular system is located in the inner ear, and it helps us maintain our balance and posture. It's something that we tend to take for granted because we don't have to think about doing things like walking or standing up. Those things are so intuitive and come so naturally to us that we really don't give it a second thought. But when the vestibular system stops functioning properly, everyday tasks can become extremely difficult. Nearly 100 million American adults have experienced vestibular dysfunction or have a vestibular disorder. Patients will experience position, movement, and balance issues like dizziness or vertigo. The current standard treatment for vestibular dysfunction is traditional vestibular rehabilitation therapy, otherwise known as VRT. Traditional VRT has been shown to be effective in treating vestibular dysfunction. It consists of a variety of rehabilitative exercises that patients will execute in outpatient care or in their own homes. However, it relies on limited observations in the clinic and subjective expert opinion, so there exists a significant need for objective quantification of patient performance in addition to these subjective measures. My research takes advantage of affordable wireless sensor technology that can quickly and accurately quantify patient head and body motion. Kinematic data of vestibular patients and healthy controls were recorded, segmented, and comparatively analyzed. Kinematic differences were revealed. The end goal of my project is to code an app that will detect patient movement abnormalities and offer feedback based on kinematic data. VRT programs can be better designed to fit patients' needs through the incorporation of inertial sensors into these programs. This will ultimately result in improved patient outcomes. Taking advantage of technology will allow for improved delivery and management of VRT programs, both in outpatient and at-home care. Developing an app based on kinematics-based analysis will hold long-term impacts on the quality and effectiveness of VRT programs both in and out of the clinic.
Hi, my name is Nicholas Pham, and I conducted research at the PRAS lab at the Johns Hopkins Bloomberg School of Public Health. And my research was centered around water disinfection, more specifically chlorination. And chlorination is the most widely used form of water disinfection because it is so cheap and effective. So the problem that I investigated within the chlorination process were disinfection byproducts. And this is when the chlorine, which is intended to kill off any pathogens or waterborne illnesses in the water, reacts with different compounds in the water and creates these unintended side products. And the group of compounds that I investigated in my research were phenolic compounds, and this is a broad group of compounds that are found in many things, such as pharmaceuticals, plastics, even food, like preservatives and flavorings. And I looked at how chlorine reacts with different phenolic compounds in the water and forms a specific byproduct called BDA, which has been proven to be toxic to humans. And I looked at how much BDA was formed from each phenolic compound, and then also I investigated the mechanisms behind how this BDA was formed to try to understand all the different steps that take place in this process. And this work is important because BDA and a lot of other disinfection byproducts are toxic to humans, so in order to successfully remove them from the water, we need to understand how they're formed in the first place.
Hello. My name is Juni Polansky, and this year I conducted research at the Schillbach Biology Lab at Johns Hopkins University. My research focused on bacteriophages, or phages, which are viruses that have the ability to infect and kill bacteria. Each phage only targets a specific host range of bacteria, and this property has made phage research very promising as an alternative to failing antibiotic treatment methods. Phages can target a specific harmful bacterial pathogen without the patient having to worry about the other, good bacteria, dying. As for my research, I worked with phage isolation. Using serial dilutions, I purified several samples of phage and created high-titer lysates. Using gel electrophoresis, I attempted to identify the cluster of each phage. I performed several DNA extractions, and I plan on sending in my results to be sequenced. For the future, I am looking into the phage-host interactions in starving conditions. Most lab research on phages has been done only in nutrient-rich conditions, which do not simulate actual environmental conditions. Looking into the phage-host interactions under nutrient deficiency will provide information crucial to understanding the function of phages in their natural environments.
Hello, my name is Tejal Schwartz, and this past year I was fortunate to conduct my research practicum at the Sidney Kimmel Comprehensive Cancer Center with Dr. Leonardo Lesnick. My topic of research is transplant immunology and is extremely important and necessary for improving the lives of people who are battling all types of immune rejection to new tissue following transplantation. This research involves studying ways to limit the body's immune system rejection response when it identifies allograft tissue as foreign and a threat to the body, and also to help patients who must undergo heavy therapy of immunosuppressive drugs in order to combat this rejection. The immune system operates on a system of self-tolerance, which describes its ability to avoid destroying non-threatening healthy cells and instead attack foreign invaders or infected cancer cells. Some patients suffer from graft-versus-host disease, where immune cells of the transplanted tissue attack the host cells or patient cells. The drug I use in my research, cyclophosphamide, is a synthetic toxic drug used to treat leukemia and lymphoma and is also used as an immunosuppressive agent. I study how cyclophosphamide affects different types of T cells and analyze T cell metabolism rates in order to manipulate the immune system response in transplant patients and therefore reduce the chance of graft-versus-host disease. During the practicum, one important method I learned was flow cytometry, which is an analytical technique used by researchers to study the physical and chemical attributes of cell populations. I was trained how to process cells and how to use a flow cytometer from my very dedicated and very encouraging supervisor. My project is special because so many people are struggling with post-transplant life, and it would be so life-changing for them if we could somehow find a means to make it so their bodies do not continuously try to reject the organs that they went through so much trouble to get in the first place. It's about helping improving the quality of life for people struggling after receiving transplants and dealing with rejection either from their own bodies or a new transplanted organ.
Hi, my research is about improving sensory neuroprostheses by measuring vibrations with a tactile sensor. There is approximately 1.3 million Americans who are living with some form of limb loss, causing many to turn to prosthetic limbs. The benefit of prosthetic limbs include more mobility and ability to perform daily tasks. When prosthetic limb is connected to the nervous system, it is referred to as neuroprostheses. Neuroprostheses allow for a more naturalistic control of the prosthetic limb and allows for sensory feedback to the amputee. Sensory signals are sent to the neuroprostheses interface in order to mimic the biological hand. The biological finger actually uses vibration to understand texture information. Therefore, we examine how the tactile sensor is available and able to measure vibration stimuli. The results from the sensor reading showed that the vibration fanned out from where the stimulation was given off, and distribution was dependent on the frequency strength. The significance of the study will show how closely the neural prostheses are to the biological hand by how vibrations are distributed to improve future neural prostheses.
Hi, my name is Jacob Thompson. I'm work at the Smith Lab with Dr. Emmy Smith and several of her graduate students, and I'm working on the coevolution of life and the environment during the Cambrian period. So the Cambrian period is a really important period in Earth's history. It began around 541 million years ago, and it's the first time that we see macroscopic life in the fossil record. So it's the first time seeing life that will be recognizable to our eyes. And so it's really important to understand how life and the environment evolved during this period, and what may have caused these evolutionary and environmental changes. So my work at the Smith Lab has focused specifically on the basal Cambrian carbon isotope excursion, which is a geochemical marker found at the beginning of the Cambrian period and is a major piece of evidence for the radiation during this time. This geochemical event is really important because it's evidence for the oxygenation of the oceans, which would have allowed for the development of larger and more complex organisms. The research question that I've developed with my supervisor and Dr. Smith is whether BASE is truly representative of global environmental changes during this time, or whether it's the product of more regional geological processes such as diagenesis. The Cambrian radiation is one of the most important evolutionary and environmental events in Earth's history due to the appearance of macroscopic life during this time. So thus it's important to study the mechanisms by which this change occurred and how the environment shifted to allow evolution like this to occur. So thus far in my research, I haven't actually been able to obtain any results, mainly because of the time it takes to process and analyze samples. But I've been really excited to learn about the various techniques and protocols that are used in my lab, especially the use of mass spectrometers.
Hello, my name is Chielota Uma, and I research at the Kennedy Krieger Institute in the Department of Motion and Physiology, focusing on the topic of reversing the effects of brain trauma with brain stimulation. Oftentimes, victims of brain trauma are left unable to do simple tasks such as writing their names or opening doors. It alters their way of living and can be extremely frustrating. My mentor and I wanted to understand whether or not transcranial direct current stimulation would be able to reverse the effects of brain trauma by allowing victims to relearn the tasks that they had lost the ability to do before. Although TDCS was an important method for me to learn and understand, we found that it wasn't as effective as we had originally hoped. However, we can continue using it as a short-term solution as we keep searching for new methods in the future.
My name is Villahermosa, and I am part of the Earth and Planetary Sciences Department at Olin Hall. My research is on Mars gravimetry and exploration. More specifically, I've been tasked to run a simulated gravity traverse on the upcoming Mars 2020 rover. This is important because gravity traverses can help us learn more about Mars's past. An important method that I've learned during my stay at Olin Hall is utilizing MATLAB as well as equations to predict what gravity would be like on certain areas of Mars.
Hello, I'm Dr. Nicole Rosen. I'm the Director of Research for the Ingenuity Project, and I have the rewarding job of overseeing students' research projects. Specifically, for 10th graders, I teach them about research methods. I help them explore and identify their research interests, and then help them find and secure mentors for their practicums. I work very closely with juniors and seniors while they're doing their independent research projects. They put their work into a formal research paper, different presentations, and then put everything together and submit for STEM competitions. And all the while, I learn a great deal from these motivated and amazing students. And our students' success would not be possible without the involvement from many different parties. I want to sincerely thank all of our mentors, all of the supervisors, parents, family members, the poly community, and fellow students who help our Ingenuity students be the best they can be. Their success is a reflection of your participation, patience, and involvement. Thank you so much. And we are always looking for new ways for us to collaborate with our community and always looking for strong role models. So if you or anyone you know is willing or interested in mentoring our exceptional students, please reach out to me. I would love to have a conversation with you. It's been a really amazing experience for me to be a part of the Ingenuity Project, to see our students grow and advance and mature over their time while they're doing their research and innovation practicums. I look really forward to seeing the contributions our students are going to make for society, as well as the collaborations that we're going to have with more parents and families and organizations and mentors as the Ingenuity Project continues to expand. It is a true treat to be working with Ingenuity students. Please don't miss out. Thank you.